Hi, my name is Dan Bittner, and today I'm going to talk to you a little bit about vesicular transport, and hopefully by watching this you'll have a better understanding about how vesicular transport works so that when you have an exam on it you can watch this video and be prepared. Vesicular transport is a process by which substances are transported from one cell to another through the use of protein-coated vesicles. And the protein coat serves to shape the membrane and to make sure that the required molecules for transport are captured. And as you can see, I have an illustration here, and it says budding vesicles wear coats, and it shows kind of how the coat proteins work when interacting with a vesicle. There are three major types of transport vesicles that transport substances through cells. There are COP1 coated vesicles, COP2 coated vesicles, and clathrin coated vesicles. So first we start with COP2 coated vesicles, and COP2 coated vesicles function to transport proteins from the rough endoplasmic reticulum to the cis face of the Golgi apparatus. And the assembly of the COP2 coated vesicles begins when SAR1 is brought to the endoplasmic reticulum membrane, which is driven by the exchange of GDP to GTP. And after the vesicle pinches off from the membrane, the protein coat is no longer needed and is shed. So next we look at COP1 coated vesicles. COP1 coated vesicles function to reorient Golgi proteins in trans to cis direction, and also to transport Golgi proteins back to the rough endoplasmic reticulum. And a retrieval protein that is associated with COP1 coated vesicles binds to proteins that need to return to the rough endoplasmic reticulum. Now we look at clathrin coated vesicles. Clathrin coated vesicles function to transport proteins from the trans face of the Golgi to their final destination. Clathrin coated vesicles bud from the Golgi on the outward secretory pathway and the plasma membrane on the inward endocytic pathway. Clathrin molecules shape the membrane into a vesicle by forming a basket-like network on the membrane's cytosolic surface. Then the protein dynamin constricts, and this causes the vesicle to pinch off of the membrane. Okay, the packaging of materials into vesicles, we have endocytosis, which is the process by which the cells take in materials through an invagination of the plasma membrane surrounding the material in a vesicle. Then we have exocytosis, which is the process by which molecules are moved out of the cell. The molecules are enclosed in vesicles that fuse with the plasma membrane and release the molecules outside the cell. So now we look at tethering, docking, and fusion. Specific RAB proteins on the surface of each vesicle are recognized by tethering proteins. The RAB and tethering proteins are specific to one another, which ensures that vesicles only fuse with the proper membrane. After the tethering protein has grabbed onto a vesicle's RAB protein, a V snare on the vesicle interacts with a T snare on the membrane. The vesicle is now firmly docked to the membrane. For fusion to occur, the snares from each membrane must come close enough for their lipid bilayers to interact. The two snares then wrap around each other, pulling the bilayers together so that fusion can take place.